Great. Yeah. Well, thank you so much for inviting me, Carota. Um, well, yeah, it's great to be here. And I kind of apologize to begin with for telling you sort of about mostly biology, a little bit machine learning, essentially very little controls, but I do hope it's going to be interesting for some of you at least. Okay, so I want to talk about sort of machine learning guided sequence design for mRNA and gene therapy. So why DNA and RNA therapeutics? Well, it's basically because it's easy to make these molecules, but we can encode like vastly complex information in DNA and RNA. And also to some extent, as we've seen for the Moderna and Pfizer vaccines for COVID, right? We know how to deliver nucleic acid therapeutics and vaccines, and to some extent, we can control immune reactions. Now, what I want to talk about is sort of this argument that, of course, when you make an mRNA therapeutic or an mRNA vaccine, right, the first thing you do is pick the protein and kind of in some ways optimize the sequence that encodes the protein. But there's all these other elements of a gene therapy vector that are just as important, in my opinion, like basically the enhancers or promoters that control location of gene expression, or maybe the 5' UTR and 3' UTR that, determine sort of the strength and also cell type specificity of expression. And so basically my question is kind of like how should you choose these other elements if you want to make a good therapy? And typically what good means is sort of one of two things. Like the first thing that people care about is simply to make enough protein or as much protein as you can, right? That's essentially when we worked with Moderna and other collaborators, that's essentially the first question they ask, like, how should we choose UTR such that you get a lot of protein? Okay. The second question that people are just starting to, in my opinion, to kind of think about is like, okay, how can we use these regulatory elements to maybe target a gene therapy to specific cell or tissue types to, for example, minimize liver toxicity? Like all of these nucleic acid therapies are going to go to the liver. If they're too active there, that could be problematic. Like, how do you stop that? So the question then is, like, how do we identify regulatory sequences with the desired properties? And I want to tell you kind of a series of stories on how my lab is identifying these sequences and then, or how my lab is kind of learning how to map sequence to function and then using these maps to generate synthetic sequences that have the desired properties. And I'm going to do this in a few different sort of regulatory contexts. So step number one is that we need to learn how to predict sequence from function, or how to map sequence to function. And I'm going to tell you a first example that's kind of an older story where we do this in the context of the so-called 5' UTR or 5' untranslated region, right? Sort of like the leader sequence that precedes a protein coding mRNA sequence. And what this leader sequence does, and this is not new, this has been known for a long time, right? It sort of helps regulate the strength of translation. So what happens is that at the very beginning of this mRNA, this so-called CAP, a chemical modification, the pre-initiation complex assembles there. So it's part of the ribosome. Then this pre-initiation complex kind of scans along the 5' UTR until it finds a start codon, the beginning of the sequence that needs to be translated, that needs to be turned to protein. Once it finds the start codon, the entire ribosome assembles and translation begins. Now, what's interesting about the 5' UTR is that there's a lot of information in that region that kind of determines how efficiently this scanning process of the pre-initiation codon, pre-initiation complex actually proceeds. So there's, for example, maybe the most or best known signal that's encoded in the sequence is the so-called Cossack sequence, right? The actual context of the start codon influences how well the start codon is actually recognized by the ribosome. Then there's sometimes or very often there's sort of competing start codons, right? You can imagine that if relative to your intended start codon. There's another one. Now you have competition that will influence how much protein is made. And then the third, maybe less intuitive element that's also has been extensively described, the sort of secondary structure. Like these RNAs kind of fold onto themselves, and depending on the location and the strength of the structure, this could be a positive or a negative thing for, translation initiation. Now, of course, these are sort of, well-known phenomena and have been described individually in many different contexts. But what interested us was to sort of ask whether we could make a model that combines all of this information and essentially just allows us to look at a sequence and score its ability to initiate translation, right? Sort of somehow we need to integrate over all of these, individually described bits and pieces. And the way my lab is going about this is essentially using sort of trying to build models through supervised machine learning, right? And to do that, we have to start by somehow generating training data. And the way we're generating these training data is very often through synthetic biology. So basically what we do here is we want to make a large library of genes that look the same everywhere except in the 5' UTR. So we're just going to sort of randomize, in fact, the 5' UTR, and we're going to generate, a few 100,000 different genes, actually, or transcripts, many more than you would actually see in the human genome, right, which supports maybe 20, 30,000 genes. We can make a few 100,000 synthetic genes, maybe just randomize the sequence of the 5' UTR and keep the coding sequence and the rest of the gene constant. Then we can essentially take this library, we can deliver it to cells. We often do this in practice in sort of cell culture. We let the cells interpret this information that we provide to them, right? So it's a few 100,000 genes. Every cell gets a few thousand of those. mRNAs are being made, mRNAs are being translated. Then we perform some sort of experimental workflow that essentially allows us to determine for each message how many ribosomes are sitting on it. And that's kind of a metric of the ability of that particular message to load ribosomes, right, to do translation. And of course, since the only thing that changes about each message is the 5' UTR, it's effectively a metric for how good a particular 5' UTR is at loading ribosomes, right? So the data set, without going into the detail of the experiments, looks something like this. It's a series of a few 100,000 5' UTR sequences, and associated with each sequence, we have a measurement of the mean ribosome load. Okay, so a number. And then, of course, we can use these data sets to train a machine learning model. Sort of originally in that paper, we built sort of a relatively shallow, like 3-layer neural network, convolutional neural network, where the network predicts ribosome load from sequence. And then we tested this network on on a held out test set. And this held out test set actually is interesting because it consists of fragments of human 5' UTRs, right? So these are not random sequences like the data that we actually trained the model on, but these are now human biologically meaningful 5' UTRs. And what you basically see here is that the model somehow from these large scale random examples, it learned the rules according to which 5' UTRs control translation initiation. And so it can predict what happens even for these, in some ways, more interesting human 5' UTRs. Okay, so now you can ask, well, what is this? Nice, but what's that model good for, right? And I think there's essentially 3 applications. I'm only going to talk about the third one really today. But the first application is essentially variant impact prediction, right? There's, 5 million base pairs that are different between any 2 of our genomes. If you look at the entire human population, there's billions of variants, maybe like every possible single nucleotide variant is out there. That's not embryonically lethal. So then determining which of these many, many variants actually, are pathogenic or have an impact on on health or even on gene expression is actually pretty hard, right? But if you have a model like this, it is very easy to, at least, for example, in this case, right, look at every possible 5' UTR variant and identify those that potentially change translation initiation. And then you can follow up on those, and understand whether that occurs in the context of a disease gene, et cetera, et cetera. But it's sort of as a starting point, I think these type of tools are very powerful. Of course, we can also use these kind of models to try and learn biology, or at least, through sort of explainable AI techniques or model interpretation, we can connect what the model learned to known biology, right? To sort of make connections. So historically, I think often people thought of these neural networks as sort of black boxes. Now I think that's increasingly less true. But the application that I really want to focus on today is using these type of models as oracles for sequence design. So making sequences that have particular properties by using this model as sort of my guide. Okay, so the second part of my presentation is essentially about how to make sequences using, once we have a trained oracle. And also, like, I don't know what the system here is, but if people have questions, I guess just jump in. So the idea of what I want to tell you is that we can essentially use, or we can learn to write regulatory sequences from scratch using model-guided design. And the basic idea is extremely simple. It's essentially just in silico evolution. Like, I can take any sequence, I can predict the fitness of that sequence according to my predictor. I can make now a change to my sequence. So here's a fitness score for this particular 5' UTR. I can change that 5' UTR. I can score it again. If in this case, my goal is to make sequences with higher fitness, I would say I keep this change, layer another one on top of it. If that again is improving fitness, I can keep it, et cetera, make changes until I reach my fitness goal, right? It's very intuitive, very simple. And I just want to say that even this very naive sort of stochastic search or in silico evolution type of approach actually works quite well. So even, in 2017, that was sort of the first time we kind of used this sort of approach of doing high throughput assays together with convolutional neural networks and generative algorithms to make synthetic sequences. What we did there is we did something similar to the experiment I described with human 5' UTRs, but actually in yeast. And what you're seeing here is, I think it's about 100 different 5' UTRs, for which we measured their fitness in yeast, and we also had a model that predicted the fitness. Then we did the exact sort of stochastic, one mutation at a time type of in silico evolution that I just showed you, to evolve all of these sequences to sort of maximal fitness. And what you can see here is that each blue dot corresponds to a green dot. So basically saying that in general, every, whatever sequence we started out with, within about 40 changes or so, we could evolve these sequences to be as fit as the best sequences in our data set. We did similar things with these human 5' UTRs that I mentioned earlier. This is a bit more general. So for human 5' UTRs, we actually designed sequences, 1,000 sequences each, targeting, every value of mean ribosome load from like 3 to 10 or something like that. So basically, a mean ribosome load of 3 is essentially a badly translated mRNA. 4, 5, 6, the higher you go, the better translation is. So what these tight gray histograms show are essentially 1,000 sequences designed to target each of these values. We then measured or synthesized and measured these sequences, and you can see that by and large, the experimentally measured data points and the model predictions agree quite well, right? Obviously, there's a little more range in the experiments, but still, the ordering of the experimentally measured sequence fitnesses is very similar to what we would expect from the model. So basically kind of suggesting that I mean, this actually was sort of a genetic algorithm, but that this type of sequence design can work quite well. And that the models that we're using to drive the sequence design actually know something about the relationship between sequence and function. Now, having said that, right, like the stochastic search, I mean, as intuitive as it is, has fundamentally has like two issues. Like one issue is that it's not very efficient because especially as we reach higher fitnesses or higher fitness scores, we make a lot of changes to the sequence that do not improve the fitness. And so there's lots of calls to the predictor that are essentially wasted. The second issue is that these type of algorithms, you know, depending on how you run them, tend to get stuck in sort of local minima or maxima. So maybe we'll generate a lot of similar sequences that are all not quite optimal. Like, so to address the first part, right, this issue of making lots of wasteful calls to the predictor, a former student in the lab, Johannes Linder, who's a very talented sort of computational biologist, developed essentially a gradient ascent approach to doing sequence optimization. So building on prior work, what he does is essentially take the discrete sequence, replace it by a continuous differentiable matrix, essentially something that looks like a positional weight matrix or something like that. And then we can do sort of backpropagation and gradient descent on this input sequence to make changes always in the direction of higher sequence fitness or higher fitness sequences. Okay. The second complementary approach, which maybe is even more elegant and general, is to just use the predictor to train a separate neural network. Right here, we can essentially do generative machine learning, where we train a generative neural network that learns to spit out sequences that have high fitness according to the predictor. Like our particular flavor of generative neural networks, we call deep exploration networks, because in addition to trying to kind of maximize fitness, we also force the network to make sequences that are different from one another. Like we basically start the network with two random seeds, and we ask it to generate two sequences that are different. They're both high fitness, but different, right? And the goal of that is to sort of explore the sequence space and not get stuck in some local minima. And also, you know, sort of, we may not entirely trust our predictors, so kind of exploring a little bit across sequence space certainly is a good strategy in this sort of bio design world. So these are sort of two different approaches, right? So gradient descent, very computationally efficient, potentially still has that issue with the local minima, and then generative neural networks, very, you know, computationally efficient once you have actually trained the network, but a little more challenging to implement. But either of these approaches, right, are potentially superior to just a stochastic search type of algorithm. Now, I just want to, you know, again, sort of give you a couple of examples of how we actually use this. So Johannes, when he originally designed this, was actually in the context of 3' UTR, so the other end of the messenger RNA or a gene. And you may not remember this, I may not have seen this for a while, right? But when mRNA is transcribed, human mRNA, it actually has to be terminated. There's a point where the transcript gets cut, and then a bunch of A's get appended enzymatically. And there's signals encoded in the 3' UTR typically that determine where this cutting and termination should occur and how efficiently it should occur. And so Johannes was essentially showing, so we built a model similar to what I've shown you before for the 5' UTR. We built the model to learn the mapping between the sequence of these polynation signals and their sort of fitness in terms of ability to terminate messenger RNA. Johannes then used this model to guide the design of synthetic polynation signals that result in different levels of termination efficiency, kind of showing that, you know, again, the model can actually generate functional 3' UTRs. But I also want to point out that the type of algorithm that we make, they're not limited to, I don't know, untranslated regions or even mRNA or DNA, but we can also use them, for example, to generate protein structures that, or protein sequences that should fold in a particular target structure according to TR Rosetta or AlphaFold or something like that. So it's pretty general design algorithms that work with biopolymer sequences. Now, since I started talking about 5' TRs, I want to briefly go back to that topic and use the next 5 minutes to tell you about sort of an extension of this initial model that we built on 5' TRs. And this is actually now, it's a Nature Comms paper that came out this year. It's a collaboration with the company 2seventy Bio. And 2seventy Bio are essentially a cell therapy company that are trying to make engineered T cells and other cell types where they knock out or somehow edit particular native genes to make the cells, better at proliferating or better at invading tumors and things like that. So the way they make these edits is that they deliver so-called megatal enzymes. So these are very highly specific restriction enzymes that, can go in, make some cut to the genome, and then that cut gets fixed with an error. That's how you make mutations. They deliver these megatals as messenger RNA, and they basically ask us to make 5' TRs for these megatal delivery mRNAs that would result in efficient translation and therefore, high gene editing. Right, so the workflow is essentially in vitro transcribe mRNA, deliver it to cells. The mRNA makes this megatal enzyme. The enzyme cuts the DNA in a specific place. After some time, we can take the cells, we can sequence, and we can ask what's the percentage of, or what fraction of genomes that we see, or how often is the particular target locus actually being edited, right? And the higher that fraction is, the better for the company, right? And so to do that, what we did is we just designed a small number, in this case, not hundreds of thousands, but just a few tens of 5' TRs for them that they then tested individually to measure gene editing. And so I want to walk you through this data quickly because it's sort of interesting in some sense because it didn't quite work the way we hoped, or at least not all of it. So on the left here, basically what you're seeing is each bar corresponds to one 5' TR that we tested. Okay, so it's one experiment with one 5' TR. There's 4 almost replicates within each group, right? These 4 actually correspond to 4 different concentrations of the mRNA that's being delivered. Afterwards, all the bars look kind of the same height because it's normalized to some reference. But you can think of these as like 4 replicates or something like that. The gold bars on the left is great. So these were sequences 5' TRs we picked from our initial library. We picked them because exactly they have different levels of ribosome loading. And you can see that, they also result in different levels of editing. The purple ones are similar controls that we picked because they have high levels of ribosome loading and they result in high levels of editing. The gray ones at the very right are actually sort of our competitors. So these are the best sequences that 2seventy Bio had to begin with. They had previously done sort of a screen of natural 5' TRs and identified the ones that were best in their context. Okay, so the first observation you can make is that like the colored bars tend to be, similar height as the gray bars, but not higher, right? So that's in some ways disappointing, but also I actually think not surprising. I'll say more about that in a second. The other thing you may notice is that, the red and orange bars, they were designed by a generative neural network. They look quite good, about as good as the controls. The green and blue bar, which we designed by this sort of gradient ascent method, for whatever reason, don't look as good, okay? Some of them have low fitness, even though they were supposed to have high fitness. Interesting. Now, of course, then we sort of ask, well, what's happening here, right? And Sebastian Castello here, who's the main author of this paper, decided that what he was going to do is look at the stability of these messenger RNAs. Because if you think about it, there's really 2 parts, 2 things, 2 features of the mRNA that determine how much protein is being made. One of them is how well the mRNA is translated. The other one is just how long the mRNA sticks around, right? If the mRNA is very stable, it can make more protein over time than an mRNA that's very unstable. And so the hypothesis could be that somehow these green and blue sequences are particularly unstable for whatever reason, right? We're not actually designing them to be stable. We're just designing them to be well translated. But maybe coincidentally, they're unstable, right? And to some extent, that's actually what we're seeing. So the experiment Sebastian did was to just, deliver these mRNAs and then do a time course, measure how much is left after different amounts of time. And then we can kind of look at the area under these decay curves and use that as a metric for the sort of the stability of the mRNA. Now, it's kind of cool and totally unexpected to me is that some of the sequences that were extremely stable, like these brown and yellow ones on the right-hand side, actually, if you go back here, correspond to mRNAs that are not necessarily well translated, right? So these are they're not they're not only not well translated, but they don't make that much enzyme. So basically, this is a functional output. We just measure how much functional enzyme is there, and how much gene editing do we observe because of that. Like here for these yellow curves, yellow data points, right? You see, they're not very good at editing at driving whatever gene expression and resulting in editing, but they're very stable. Right? So it's interesting. So they're badly translated, but super stable and result in some sort of intermediate or low level of editing. The sequences that are really good, the red and orange and purple, right, sort of in this scale have sort of intermediate stability, but are very efficiently translated. And then the sequences that kind of failed are still fairly highly translated, but become less stable. But it's this very non-linear relationship between sort of stability, like between editing, like the actual protein output, stability, and ribosome loading. And all of this to say, I mean, I think this is actually still remains a very interesting biology problem, trying to figure out, how these different quantities actually relate to one another, and eventually learning how we should design, or with what kind of objective function we should use to design sequences that result in, that maximize the particular thing that we actually want to maximize, which in this case is gene editing, as opposed to ribosome loading, which is what we actually optimized for. So just to end on a high note, I do want to say we sort of did the second generation of these designs where we actually just shortened the 5' UTRs a little bit, and we're able to, get much more robust behaviors, even using just, trying to design maximal ribosome loading, and even identified some sequences that performed well relative to the controls. Nonetheless, I think fundamentally, I mean, there's sort of a limit to how much we can achieve by engineering only the 5' UTR. And I think it just has to do, I mean, in my mind, at least my mental model now is that basically what these 5' UTRs do is a lot of negative regulation. Like there's sort of a perfect 5' UTR, maybe the initiation is actually limited by recognition of the cap or something like that, like how often the pre-initiation complex can actually latch on to the very beginning of the message. And then the rest of the sequence, in the best case, just get out of the way and these pre-initiation complexes slide through. And anything you do to the sequence essentially makes it a little worse. And that's why it's hard to actually design sequences that are, vastly superior to what we find in biology. But nonetheless, I mean, I think that's a very interesting, understanding that, I think, to me was at least interesting. Now, the next thing I want to get to is kind of asking about cell type specificity, right? So I mentioned at the very beginning that it's essentially two goals that people have when they engineer mRNA, or when they ask, why should I make a particular 5' UTR or 3' UTR? The first goal always is like, let's make a lot of protein. Okay, that's what we've been trying to do here. And probably we can do it as well as anyone. The second question is kind of like, well, can we target protein production to one cell type or another, typically to minimize side effects, right? And so one question you could ask is like, well, could we do this at the level of translation? In these assays, we just tested, our editors in a couple different cell lines, and essentially found that editing efficiency, it's sort of noisy data here, but editing efficiency is quite correlated. We then actually did a bigger screen of hundreds of thousands of sequences across 3 different cell types, including primary cells, T cells, and again found that editing or that translation ribosome loading in this case is very highly correlated, which again, I think this is not necessarily true. I'll show you an example of that in a second in general, but it's probably true for these sort of like happily dividing, rapidly growing cells. The counterexample to some extent, but not entirely, right, comes from this paper that we recently, or that we're just about to publish with Alex Shear's lab and Madalena Madeira Raimao Pinto, who's the first author of this work, where we actually did sort of a massively parallel reporter assay. So again, a large library of 5' TRs in front of a reporter gene, injected in a zebrafish embryo. And then we let the embryo sort of develop for a while, like divide and grow, start differentiating, and collect the ribosome-loaded mRNAs at different time points to kind of see whether, particular sequences get translated differently at different stages of development. And in fact, we do observe that kind of behavior for some of these sequences, which kind of, again, suggests that there's definitely some level of regulation of mRNA translation across sort of cell states and cell stages or cell types. Nonetheless, in our hands, it's not, it wasn't easy to identify regulatory elements that would like just entirely target translation to one cell type versus another. And so that kind of then brings up the question, how do we engineer cell type specificity? Okay. And so that's the next 1 or 2 topics, depending on time, that I want to talk about, is like kind of how can we actually make mRNA therapies first and then gene therapies that result in that allow us to target gene expression to one cell type or another. Okay, so this next vignette is actually work by Lucas Oesinghaus, also a very talented postdoc in the lab. And what Lucas decided to do, and this, I should say, is a very old idea, but it's a very good idea, is to take advantage of microRNAs to try to destabilize messenger RNAs in specific cell types, but not others. So microRNAs, for those of you who don't know, are short, like 20, 21 nucleotide, long regulatory RNAs that occur in eukaryotes, human, etc. Okay, they're typically the single-stranded, they're part of a big protein complex, the RISC complex, as shown here, that's this like yellow egg, and they can bind to complementary or partially complementary sequences in an mRNA. It doesn't have to be the 3' TR, but often is the 3' TR. And if the complementarity is high, then the microRNA can effectively cleave, or the RISC complex effectively cleaves the mRNA and completely degrades it, right? So this is for an engineer, I think this is a very appealing mechanism, because I can just, if I know the sequence of my microRNA, I can just stick a complementary sequence into my mRNA reporter, and I can have it removed from anywhere where that particular microRNA is made, right? So it's very predictable and engineerable. Now, the other aspect of microRNAs that is sort of desirable is that they do exhibit some degree of cell type specificity. Like, for example, here, if this orange cell is a liver cell, there's a well-known microRNA mR122 that is very highly expressed and very specifically expressed in liver. So many people for many years have taken advantage of that kind of specificity to make reporters, right? For example, here by including multiple copies of a complementary site for mR122. If this gets delivered to the liver, the mRNA will probably get destroyed. Anywhere else where the microRNA is not made, the mRNA will survive. Okay, so that's the basic idea. So the reporter activity on the right here is sort of showing that this reporter should be expressed or should be translated and stable everywhere except here in this yellow-orange liver cell. Okay. Now, the interesting question that Lucas asked is like, well, what other patterns are possible, right? Like sort of this one situation where you exclude expression from one particular tissue using one very specific microRNA, it's very convenient, but may not be what you always want. Maybe you want to do the opposite. You want to have expression only in the liver and nowhere else. How would you achieve that, right? Like the naive answer might be to say, okay, find a microRNA that's specific to every single cell type where you don't want the expression, then just concatenate all these target sites into the 3'UTR. Two issues with this. Biggest issue is that these microRNAs generally don't exist. There may not be a highly expressed, very specific microRNA for each cell type. Second issue is that even if they did exist, like if you had to put together like 20 of these target sites in a 3'UTR, it'd probably become unstable for other reasons. So that doesn't work. So how somehow can you do better? Okay. Now the first thing, and this is kind of interesting for people who work on microRNAs anyway, this is the first thing you have to, if you want to engineer sensors for microRNAs, you have to know what these microRNAs levels actually are in the different cell types. And when Lucas started looking at the literature, he found that different reports or different measurements of microRNA levels absolutely do not correlate with one another in general. And this is really an artifact of how these microRNA levels are measured and like artifacts of sequencing library preparation, et cetera, et cetera. So the first thing he actually had to do was to kind of go through all these data sets and find a subset that did correlate well and that we could sort of use as a ground truth for the microRNA levels. Then he decided to kind of, measure, try to map out the relationship between microRNA levels that we now believe we know and reporter activity across many cell lines and many microRNAs. So he built a library where he took, the 1,000 or 2,000 most common human microRNAs and inserted target sites into a reporter construct. So very similar to the 5'UTR story, right? So now we're varying the 3'UTR, we're not randomizing it, but we just put target sites for microRNAs. But again, make a big library, deliver the entire library to cells, use sequencing to count the mRNA abundance. And then we can say, right, for a microRNA that's very effective, that's highly expressed, it will destroy a lot of mRNA. So we don't expect a lot of the reporter mRNA with that target. If a microRNA is not present in a particular cell type, then we expect that reporter to be, mRNA to be stable, and we see it a lot. Right? So we can basically just look at mRNA counts, maybe normalized to the DNA input to determine the stability of, or the impact of them, of the microRNA. And what Lucas found, and this is really kind of still, I find very surprising, is that there's this beautiful universal relationship between microRNA levels and reporter activity. Like what you're seeing here, this is actually just 100 or 200 human microRNAs across 4 cell lines, but the data looks exactly the same if we do 10 cell lines and 1,000 microRNAs. Each data point, each little colorful dot here is one microRNA measured in one cell line. Now, because the same microRNA could be made at different levels in all of these 4 different cell lines, right? There could be 4 dots for a particular microRNA. And because there's a few hundred microRNAs, there's, many dots for different microRNAs, different cell lines. But all of those essentially follow this dashed curve, which is just a model that is basically 1 over 1 plus x. It's a Hill function with cooperativity n equals 1. That's a super simple model of microRNA regulation. And the thing that's still kind of amazing to me is that this seems to hold more or less across all microRNA sequences, independent of the identity or the microRNA, or even independent of like any major cell type specific effects. So this is cool, right? Because unlike in the 5'UTR, where we sort of learned this, deep convolutional complex neural network relationship between, or used a neural network to learn the relationship between sequence and function, here we actually have a very simple sort of biophysical model that relates, the basically just microRNA abundance to mRNA stability. And then the second thing we need to ask, of course, is like, well, in the previous slide, I showed you what happens if you have one target sites for a microRNA. But if we want to realize more complex functions, we need to have multiple target sites. How do they interact with one another? And what these data on the right tell you is that we make a model where we assume that each microRNA acts independently and that concentrations are effectively additive. Like if you have microRNA 1 at some level and microRNA 2 at the same level, it's the same as having either of them at twice their level. And so it's a very simple additive model, seems to work very well. And so now we can actually go back to this original question I asked, now I have a model that tells me how a microRNA regulates an mRNA. I can even say what happens if I combine multiple microRNAs. Now I can go in and I can just try to find combinations of microRNAs that are best at approximating a particular, function that I want to achieve. Like, for example, targeting gene expression to the purple cell type, but not other cell types, etc. And so the first thing that Lucas did was just to show that he can essentially make sequences that exclude gene expression from, one cell type at the time. And in this case here, he uses up to 5 microRNA target sites in the 3' UTR, and then just sample through a bunch of different combinations to identify that combination of microRNA that is most close to the target behavior, right? And these are experimental data, and you can see it's not perfect, right? There's some cases, here, HUH7, that's again a liver cell line that makes MER122. So actually here the model sort of found the trivial solution works quite well. In other cases, like in this is one design here that targets MCF7, but you can see that it has some sort of off-target, repression in a different cell type, right? In some cases, it's simply not possible to identify the right combination, but in many cases, it is possible to identify combinations of microRNAs that achieve this function. And we can also do the much harder problem of trying to do the opposite. You can see that it works less well, right? The diagonal is not completely white as one would want for high activity here, but still, it works maybe better than I expected. And so he kind of went through and experimentally designed tested a bunch of different designs targeting inactivation in 1, 2, or 3 cell lines, or activation in 1 or 2 cell lines. And in general, this works quite predictably, which I find actually kind of find astonishing. Now, okay, actually going back to here, right? So it works very well, but it's nowhere near perfect. And I think there's essentially 3 reasons for that, right? The first one is that microRNAs are only negative regulators, right? So we cannot do things like, activate somehow translational stability in one cell type or another. So all we can do is exclude, which is sort of limiting. Second is microRNAs are weird in the sense that they cannot regulate themselves. So a microRNA is actually made in the nucleus of a cell, and it's active in the cytoplasm. So the microRNA cannot go back and sort of regulate its own gene to make like multi-layered circuits. And then third, maybe the biggest issue is that this microRNA regulation, as I've shown you, is essentially, there's no nonlinearities that would allow you to make sort of really nice, like switch-like behaviors, right? Now, so these are disadvantages. The advantages, of course, are still in the context of a messenger RNA, you may not have many other choices, right? Like the fact that you can actually just predict these interactions accurately based knowing just, the sequence allows you to make a link to the target, and then the concentration allows you to predict the impact on the target. I mean, that's kind of amazing. And I think for mRNA therapy, that's pretty much all we're going to get. But if we look at gene therapies, right, then of course, we have a much wider toolbox of engineering, of elements that we can engineer, right? And in particular, if you ask about cell type specific gene regulation, I mean, fundamentally, this is happening at the level of transcription, right? Most people who study cell type specificity essentially will focus on like transcriptional regulation. And so, of course, you could also ask, well, if we work with gene therapy where we need promoters and enhancers and whatnot, could we engineer regulatory elements at the level of transcription that result in cell type specificity? And so just very briefly, right, to introduce this concept of an enhancer, enhancers are essentially cis-regulatory sort of DNA sequences that are often relatively far away from their target gene. These enhancers bind transcription factors, like shown here. And then if the transcription factor is bound, somehow some interaction between the enhancer and a cognate gene promoter can occur and drive expression of a target gene, right? If in this other cell type, a different enhancer is bound by a different transcription factor, this will activate different sets of downstream genes. Okay, so that's essentially how enhancers, control cell type specific expression. Now, this last brief project I want to tell you about in 3 minutes or so is due to Chris Yin, who's a very talented graduate student in the lab who essentially decided to make cell type specific enhancers. And he was going to use, or he started out using a data set for model training that already existed. So it's from this paper by Ernst et al., where they screened a few 10,000 human genome fragments for their ability to act as enhancers. So the way they set this up is shown here. So they take these enhancer fragments and they stick them upstream of so-called minimal promoters, so a promoter that's very weak in the absence of an enhancer. And then they use this construct to drive a reporter gene, which contains a barcode. Now, each barcode, basically just a unique RNA sequence or DNA sequence, is associated with a specific enhancer, such that we can use the abundance of this barcode as a metric for how good the enhancer is at driving gene expression, right? And we can measure that in one cell type and a different one, and we can look at the ratio of the strengths of these for each barcode to kind of determine how good the particular enhancer is at driving cell type specific expression. So Chris essentially took these existing data, trained a model. Model isn't that great, you can see here, but it's as good as it's going to get because that's similar to the interreplicate correlation. He then combined this model again with the same sequence design algorithms that I've introduced earlier, like the gradient descent method and the generative neural network method, and designed sequences that target one or the other of these two cell types. Okay, and the color here, so blue versus orange, tells you what the target cell type is. Like the blue ones should be in the upper left corner, the orange ones in the lower right corner. And these are experimental measurements, right? And you can say that's worked reasonably well. Like especially for the orange, it seems like, the most of the sequences are, highly specific to that HepG2 cell type. The blue one, at least they're above the dividing sort of the axis diagonal. There may be not as many as we would like in the upper left corner. Nonetheless, maybe that's okay, right? Because the cool thing about these type of experiments is that, of course, we can now take these data that were measured in the same, general context as the original data, and we can just use these data, even if it's just a few thousand data points, to fine-tune our model. And we can then use the fine-tuned model together with the generators to make a next generation of sequences, right? Sort of through iterative learning. And what you can see here, and I really kind of like this contrast, right? So this was generation 1, this is generation 2, right? So you can see now that almost all the sequences that we designed now actually work, really fall into the parts of the specificity space where we want them to be, right? And so this is not quite active learning in the sense that we didn't really try to sort of maximize or look at the region where the predictor is particularly uncertain, but it's sort of iterative learning where we basically just tell the predictor about a part of the sequence space or the activity space that we actually care about. And we give it even just, a few hundred, few thousand examples of high quality performance. And that was enough to kind of retrain it and allow us to kind of, very reliably generate high performing sequences. We can also make enhancers that are very short, which is sort of interesting and remain specificity. We can interpret, of course, again, look at why a particular enhancer is good at driving cell type specific gene expression, or like which motifs, which transcription factor binding sites are actually occurring in these sequences or are enriched, either in the design sequences relative to the initial ones, or in one sequences specific to one cell type relative to the other. And I think that's interesting just in terms of understanding why these sequences work. And I think in the interest of time, I'm going to skip these last few slides where what I wanted to say is basically that, of course, the next thing we have to do is we have to go beyond, making these models on just a few cell lines, but we have to generalize to tissues and cell types that are in the human body for which we maybe can't do these high throughput experiments. And so one of the things we're currently doing is sort of engineering cell type specific enhancers, starting not from activity measurements, but from DNA accessibility data. And that generally seems to work well, but I'm going to skip this part in the interest of time. And when I just sort of summarize what I told you, again, apologize for the lack of control, but hopefully there was some interest in terms of biology and machine learning. So what we do in my lab, right, is we essentially create these high throughput data sets where we read out the fitness of a large number of sequences using some sort of trick relating to high throughput sequencing. We then train models on these data sets, right? Sometimes these models are machine learning models. Almost always they're machine learning models. But sometimes, as you've seen in the case of the microRNA, these are actually biophysical models that we can train on these data or learn from these data. And then we use these models to guide the design of synthetic sequences that have desired properties like high translation rates, high stability, cell type specific transcription or stability and whatnot. And of course, all of this in the end, right, we believe really will help in making better, sort of not only help our understanding of the underlying biology, but also result in better mRNA therapeutics and vaccines. And I guess with that, I just want to, thank all the wonderful people I have in the lab or who are working with me, and collaborators from various companies and universities. And well, thank you all for, hanging out and hopefully listening. And again, hopefully it was interesting, even if it wasn't control theory.